All right, so our last speaker for the developmental core group is one of the 2 faculty recruits that you heard about earlier. So Dr. Burt is, whoa. Well, I have here a new faculty recruit. I know neonatology, so come on up. Human fetal T cell development, implications for tolerance, infection, and vaccination.
Thank you very much, and thanks for the chance to speak here today. I'm really excited to finally be at Duke and to be part of the CFAR. So you may be asking yourself, why is a neonatologist here talking about fetal T cells? And hopefully by the end of the presentation that won't be such a mystery. I did my early training with Mike McCune at the Gladstone Institute and at UCSF, focusing on HIV pathogenesis and the role of endogenous factors like apolipoproteins and immunomodulatory enzymes. And then took a bit of a sojourn into developmental biology, and I think the two actually come together to raise some important questions about infection and about potentially about vaccination in the newborn. So my lab focuses on development and function of the fetal and neonatal immune system, and we've taken several tacks in that arena. Recently, really focusing a lot on molecular and omics studies of the cells themselves. But I also want to focus on this aspect that I'll tell you about today, which is really a more translational aspect. And the 3 points that I want to tell you about are. To leave you with are that the human fetal T cells have distinct functional and transcriptional characteristics compared to adult T cells and are adapted to the unique developmental needs of the fetus and specifically towards generating tolerance. The neonates are in a state of transition from this fetal program to a postnatal or adult-like immunity, and so I sort of use those two interchangeably, even though they're not clearly the same. And there is a natural variation in the extent of transition, and that this transitional state may have implications for several aspects of neonatal and lifelong immunity. This question was raised in the context of another huge interest of mine, which is preterm labor and the immunological basis behind preterm labor. As a neonatologist taking care of preemies, this is something that's really near and dear to my heart. But this is a famous quote from Peter Medawer that kind of summarizes the question, which is, how does the pregnant mother contrive to nourish within herself for many weeks or months a fetus that is an antigenically foreign body? And then this can also be rephrased, why does the mother not reject the fetus, which is antigenically half foreign? But I think just as puzzling and just as interesting is, why does the fetus not reject the mother, and taking a more inside-out view of the question. And I think there's been an assumption in immunology and developmental biology that the adaptive fetal immune system is undeveloped or inert or ineffective, leading to a model something like this, where not much is going on during gestation, but that at birth this deluge of antigenic exposure leads to the acquisition of immunological experience and memory throughout life. However, the challenge that I'd like to raise to the assumption, and many groups are doing this now and have really contributed to this field, is that the adaptive fetal immune system is instead highly active and carries out the crucial task of achieving tolerance to not only self, but to the maternal environment in utero, and potentially towards other antigens encountered in utero, such as pathogens. And just looking at the actual timing of classical T cell development or alpha beta T cell development in human fetal development, by 5 to 6 gestational weeks, hematopoietic stem cells are found to colonize the liver, which we think of as being true hematopoiesis at that point. At 8 to 10 weeks gestation, we see the emergence of alpha beta T cells in the thymus. And by second trimester, we see a very robust population of mature T cells in secondary lymphoid organs, including the spleen and lymph nodes. And in parallel, and I'll talk a little bit about monocytes, but not too much today, monocytes and other sort of non-adaptive to quote unquote, cells are abundant in several tissues by mid-second trimester, evolving in parallel with the T cells. We looked initially, and this is going back to when I was a postdoc in Mike's lab, at just regulatory T cells and Tregs populations in fetal tissues, and this is looking at fetal lymph node and fetal spleen. And what we see is that these cells, which are marked by CD25, which is the high affinity interleukin 2 receptor, and FOXP3, which is the driving canonical regulatory transcription factor behind these cells. These cells marked here in the upper right quadrant are highly abundant in fetal tissues. And how does this compare to postnatally? We did those experiments and found that compared to adult cells, the fetal tissues were packed with these cells at a higher frequency. We looked at this by both flow cytometry and immunohistochemistry, histochemistry, sorry. To summarize some experiments that we did, this does not appear to be due to increased thymic output of natural T cells, but rather from peripheral conversion of other T cells into these regulatory T cells that are important for generating tolerance. And this is an in vitro experiment where we took naive T cells, eliminated the regulatory T cells from them and stimulated them with a third-party antigen-presenting cell. And you'll see as we go through this time course from left to right that the fetal cells on the top really acquire a very clear population of regulatory T cells, or Tregs, as I'll call them, compared to cells taken from adult tissues. And that these cells, if we actually do functional assays on them, are suppressive. And this is really the sticking point, that these cells are not just phenotypically Tregs. They are able to suppress immune responses against not only self-antigen, which we think of as being the really important job of these cells in utero and throughout life to prevent autoimmunity, but really to maternal antigen. Really, and the importance of that is that it speaks to the ability to acquire a specific tolerogenic response in utero. The nature of these cells seems to be programmed at the level of the stem and progenitor cells, and this is summarizing an experiment in which we used hematopoietic stem cells from either fetal bone marrow or adult bone marrow, matured them in a humanized mouse model, the Skid-Hugh-Thigh-Liv mouse model that allows for full lineage maturation of T cells in a humanized mouse. And you can see that the T cells, when we look at their final phenotype, have a high abundance of these regulatory T cells. Transcriptionally, they also look like primary regulatory T cells from the fetus. So there seems to be something programmed at the level of the stem cell during fetal development that leads to this important phenotype. This is looking at that primary signature that I mentioned, and I'll talk a little bit more about this as we go forward. But there are clearly transcripts that were enriched in phenotypically identical cells. So we looked at adult naive T cells, adult Tregs, fetal Tregs, and fetal naive T cells, and found groups of genes that were expressed uniquely in either fetal lineage or in adult lineage. And so transitioning to the newborn, what happens, what is the state of the newborn is a question that I think becomes clinically quite relevant. And the newborn state likely represents an intermediate state between this tolerance-producing fetal state and the postnatal or adult-like state, which is geared towards fighting against pathogens that can cause disease. And so you can consider two scenarios that may lead to alterations in sort of the mean state of the newborn. One being a premature maturation from that fetal state to the adult state, and one being a delayed maturation. And these have some implications, and can be broken down into two parts. That the newborn represents a transitional state between fetal and adult immunity, and that there's natural variation in the extent of this transition at the time of full-term birth. And the implications of a precocious maturation, which can be kind of depicted as this overrepresentation of the adult-like or protective immune state could be that these infants are more able to fight classical infections. On the flip side, could these infants be more prone to autoimmunity due to impaired tolerance? And if we think about the opposite scenario in which there could be delayed or prolonged fetal to adult transition, oops, sorry. Oop, what did I do? There we go. We asked the question, are these infants more prone to infection because they are more likely to tolerate infections? And could these infants be more prone to poor vaccine responses? We know that infants in general are susceptible to poor vaccine responses requiring multiple boosts. And so there could be, and we know that some infants, in spite of being boosted, don't have as robust a response. On the other side, again, could these infants be less prone to autoimmunity? So to get at these initial questions, we did some pilot work, and the hypothesis behind it was that the degree of transition from fetal to adult-like immunity could influence the nature of the immune responses. And the approach that we took was to generate a signature score. So we sorted the naive T cells, just given that those were likely the most nascent from the thymus and most representative of an unmanipulated or unstimulated population. We also looked at classical monocytes for reasons I won't discuss, but I'm happy to discuss afterwards if anyone's interested, because they do represent the other branch of immunity, which is crucial, the innate branch of immunity. And we specifically looked at these in peripheral blood to eliminate any questions of tissue residence versus circulation, and carried out transcriptional profiling, and were able to derive a bulk signature score. And I use the word bulk intentionally, meaning that these were scores based on a population of cells lysed with signal averaging. And that also becomes important in further research in which we do single cell work that I'm happy to talk about also. So we use the signature, which we derive to represent the fetalness or the adultness of a population, to ask the question, to what extent does persistent fetal or adult signature correlate with infant immune responses? Again, the hypothesis that the fetalness of the immune system will correlate with certain clinical outcomes. Don't worry too much about these. I'll show you these again and go deeper into them. But the approach was really to look at umbilical cord blood, and this is where the use of peripheral blood becomes important because we are looking at circulating cells. We sorted out naive T cells, CD4 T cells, and monocytes, and used a multiplexed qRT-PCR platform from the Fluidigm excuse me, company, and carried out a signature analysis on samples. The goal of this is ultimately to correlate with clinical outcomes. And I'm going to show you some, and I preface this by saying that these are really pilot and validation work that I'm going to show you, and hopefully to generate some interest in using these technologies to ask clinical questions in more carefully and curated cohorts. This is a quick look at the signature that we did derive, and we asked for a common signature. We asked for a signature that was common in T cells and monocytes, and the reason we did that was to get at a basic hematopoietic signature and therefore eliminate cell type effects, hopefully. And we were able to identify genes that met our criteria for significance, but also fold change, and those are in red on these plots. And you can see the CD4 T cells are shown here on the left and the monocytes on the right, and genes that are either up or down in the same direction in both cells were identified. There were, of course, just interesting genes biologically that were unique to either monocytes or CD4 T cells that were up or down in those cell types. We then used principal components analysis to understand how important each of these genes was to the fetalness or the adultness identity of the sample. And we're able to assign a weight to that gene, and that's what the plot on the right represents, the length of the bar indicating the weight that was given to the gene. And then getting to the actual samples, we looked at a cohort of healthy newborn cord bloods and asked whether simply there is this variation that we predict. And we do in fact see that in T cells on the left and in monocytes on the right, there is quite a spread of scores. And in the mathematical setup for this plot, a highly positive score correlates with an adult-like score, and a highly negative score correlates with a fetal-like score. And these are samples, control samples. And you can see that if we focus on the T cells, there are samples that do fall within that fetal-like range. Similarly, there are some samples, and these samples are individual cord samples that seem to be farther on the adult-like range. So that did tell us that there does seem to be natural variation. We were able to work with collaborators who I'm sure folks in this room know, Deb Prasad and Yvonne Bryson, to look at a cohort of kids throughout the first 7 months of life. And we do see this maturation from 0 to 120 days of age in the signature score. And what is frustrating to human immunologists, but is reassuring to us, is that there is this variation that does persist throughout the period that we're studying. And if we look at gestational age, looking from late preterm at 33 weeks, and this again is still cord, to post-dates at 42 weeks, we do see variation at any given date in the score, but we also see a positive predictable maturation across the cohort. And this is looking at an asthma cohort, actually, the Eureka cohort out of Madison, Wisconsin. So here we get to some very pilot data to ask the questions that I think are quite interesting, which are whether the score could correlate with or could predict poor vaccine responses. And the hypothesis again is that a more fetal-like score would predict a poor vaccine response. And we worked with collaborators who work in South Africa to look at BCG vaccination, and also again worked with Yvonne and Deb to look at their cohort in South Africa and Brazil of perinatal HIV infection, predicting that a more fetal-like response could result in less immune activation and therefore less likelihood of transmission vertically. And so this is some data from the infant cohort asking about the effective maturation, and this is the study design that they are carrying out, which is to obtain samples at birth, or importantly, within 48 hours after birth. So this becomes a distinction between cord again and peripheral blood of the newborn. And then to look at primary immune responses around 1 to 2 months by antibody ELISA or T cell responses, and then again at a year by antibody ELISA and to evaluate cytokine T cell responses. And this is feasibility, comparing cord samples in their cohort with PBMC samples in their cohort. Some of them paired, and where there's a line between samples, this is a paired set of samples, and they're very few. But we can see that there does appear to be somewhat of a trend. There was not a statistical significance in this very small, albeit cohort. This is feasibility data showing that they are able to parse out low responders and high responders to the BCG vaccine. And this is a cytokine ELISpot assay looking at any of interferon gamma, TNF alpha, or IL-2 response to BCG antigen. This is some feasibility pilot data from the vertical transmission cohort, and again, very small numbers, but the HIV exposed negative samples are in the black dots. In utero, HIV infected samples are in the blue squares, and intrapartum HIV infected samples are in the triangles. And again, these are suggestive trends that here the HIV exposed uninfected infants do seem to have a lower or a more fetal-like score. Certainly nothing to hang your hat on, but tantalizing to potentially ask in with larger numbers. And so in the last couple seconds, another really interesting question is to ask whether this fetal tolerogenic response could persist into adulthood, and what would be the implications of that. This could be either because of persistence of the adult or the fetal stem cells that lead to the progeny cells, or the program itself in the progeny cells in long-term progeny. Does an overly dominant fetal immune response predispose to tolerance of tumors, for example, in an adult or in an older child, or does it protect from inflammatory responses? Just as interestingly, are fetal hematopoietic stem cells involved in hematopoietic reconstitution after non-ablative chemotherapy, where one's own immune system is recapitulated by one's own stem cells, or after stem cell transplantation? And this has a special, I think, special implications for cord blood transplantation, which we do very commonly now clinically. And then a question I've always been quite interested in, but haven't looked at yet, is whether reconstitution of the T cell pool after initiation of antiretroviral therapy could be measured to understand whether some patients reconstitute with a more fetal-like population. We know that repertoires are certainly shifted after profound ablation of the T cell pool, and so I think that there's good rationale to ask whether the nature of the cells that come back could be developmentally different. And then also, could the fetal lineage be intentionally reawakened in adulthood in order to generate tolerance? And this has been the focus of a lot of our molecular cell biology work. So there's a lot of people to thank going back several years. And so I will take questions if we have time, or afterwards.